Hello everyone, my name is Cassie BAIRD, and I will be discussing the process of stem cell culturing, what it is, and the clinical relevance in a medical and microbiological setting. Stem cells are pluripotent, meaning they are the cells in our body that can grow or differentiate into any type of cell. So rather than differentiating into only skin cells or only liver cells, they can be driven to differentiate into any type of bodily system's specific cell. Three major groups of stem cells that we will be working with in the laboratory setting are embryonic stem cells, adult stem cells, and induced pluripotent stem cells. The differences between each category largely depend on where the cell was harvested from. Like other cells, stem cells grow in culture and require some of the same cultivation processes as the other cell groups. Frozen stock must be thawed and the cells must be plated, but the media must be changed periodically as stem cells are cultured or grown in continuously, and they grow in colonies. Module 5 depicts eukaryotic cell manipulation and cell culturing, which are two topics that directly relate to culturing stem cells, but culturing stem cells that can be used in a variety of experimental processes also relates back to other modules that have been discussed throughout the course. Creating a reliable cell colony that can be manipulated for a variety of experimental purposes opens the door for other procedures to be used, such as PCR and Western blotting, that are capable of measuring results for the desired experiment and answer other hypothetical questions that may have been posed by the author. Along with any other biotechnology, stem cell research poses some limitations and concerns. One of the largest limitations surrounding stem cells is the ethical concern for how they are harvested and if cloning is morally okay. Other laboratory limitations include the high risk for both bacterial and viral contamination, as well as the genetic stability of the cell lines that are being created. Despite the previously mentioned limitations, there are several advantages to using stem cells in research. For starters, stem cells are capable of morphing into any cell tissue in the body and are genetically identical to the parent cell. This makes replicating results much easier and more reliable. Stem cells show much promise for the future of regenerative medicine as well as aesthetic medicine, which would be beneficial to a large variety of patients suffering from different ailments, different diseases, and even insecurities, as the regeneration from the aesthetic point can help with their appearance as they are recovering from illnesses that may have altered that previously. A different form of stem cell research that we are all most likely familiar with, even if we don't realize it, is somatic cell nuclear transfer. This form of cell cultivation was popular all the way back in 1996 at the very beginning of stem cell research. The part that we are most likely all familiar with is Dolly the sheep, as this is the method of how she came to be. It works by taking the cell from an individual and transferring it to the nucleus, where the DNA is housed, and then taking that and inserting it into the egg where it can then grow. Thus, another name for this method is reproductive cloning. Different chemical factors are used to drive or encourage stem cells to differentiate into their desired target cell, but there is no direct control that is utilized during this process, as it is only meant to grow the cells, rather than to create experimental conditions. This figure is from the paper Allogenic Cell Therapy Using Umbilical Cord MSCs on Collagen Scaffolds for Patients with Recurrent Uterine Adhesions, and it is a phase 1 clinical. So this is immunohistochemical staining before and after treatment. The pre-treatment is the above slides, and the post-treatment, 3 months post-treatment, is the bottom slides. And it shows a differentiation or upregulated differentiation, proliferation, and neoanagenesis of the different types of cell cultures after treatment with stem cells. So, in summary, it is showing the ability for stem cells to create a healing and improved state of treated cells. This figure is from the paper Clearance of Apoptotic Cells by Mesenchymal Stem Cells Contributes to Immunosuppression via PGE2. And this figure is showing how stem cells are able to increase the clearance of apoptotic cells and increase immunosuppression in the SLE after treatment of stem cells in tissue culture. This figure is from the paper Stem Cell Enriched Lipotransfer Reverses the Effects of Fibrosis in Systemic Sclerosis, and it shows more of the aesthetic side of stem cell medicine. This figure specifically shows the improvement in vertical furrows and soft tissue bulk after the stem cell enriched treatment. This shows the potential for stem cells to aid in the healing process of cosmetic procedures and further emphasizes their importance in regenerative medicine as a whole. Thank you for watching, and I hope you learned something interesting about stem cells today.